Hello, good afternoon. Here we are on Thursday. I got this wrong on my last class. This is May 7th. I said April in my last class. So today what we're going to be doing is we're going to be reviewing DNA, and in particular we're going to be reviewing mostly DNA replication. One of the three big steps, the central tenet of molecular biology, arguably one of the most important things for us to really have a good grasp on going into the AP exam, which at this point is coming up pretty rapidly. Remember, next week will be our last week for review and to reiterate material and to get the practice that we need before test time. So, if you guys take a look in your folder, we have the DNA review PowerPoint first. That's what I'll go over. The DNA replication review is literally a single image. Just a single image that you guys should be able to label some of the parts of. The note set that you see here at the bottom is totally separate, and that is notes from another AP teacher for what they give their students. They were sharing them in one of our Reddits, and they invited everybody to borrow and use whatever portions of it. So I will warn you, for some reason in the note set, they don't refer to things as nucleotides. They refer to things as nucleosides, and I don't understand that. That's not me. My guess is that it was just a misspelling that continued through, but it happens like a lot. All right, so let's talk about DNA then, and let's go over exactly how replication happens. Now, I've gone through and I've kind of pared this down and added some other things in there. I'm only bringing this up because it's really important to remember what the simple piece is and what the complex piece is. And keep in mind that the complex piece, when we're talking about a situation such as this, is the complexness of it is the DNA as a whole. The monomer, the pieces that make it up, that is going to be the individual like nucleotides that are involved. And we talked about condensation versus dehydration synthesis in does it take water away to make a bond or does it break a bond by using water? All right, so let's review our actual DNA and RNA. So you guys should remember that they are individual nucleotides. For instance, what we're looking at right here in this section, that is just the adenine that we see over here that's linked across to the thymine. It's pointing out the different groups, our phosphate, the sugar, which remember in RNA is ribose, which is different from DNA, and obviously our nitrogenous base. And the nitrogenous base that you see here, that's going to be adenine, thymine, cytosine, guanine, or uracil. That's the rung, as it were, of the DNA chain. Now, the bonding, we went over the other ones. Remember, for lipids, the bond, those are going to be our ester bonds. When we're looking at proteins, those are polypeptide bonds. When we're looking at DNA, it's phosphodiester bonds. Hopefully, pretty easy to remember because this is really the only one of our biomolecules that has phosphorus or a phosphate group. Phosphate group, phospho. All right, remember the difference between them largely comes down to just the fact that RNA is single stranded. And that single stranding gives it a lot of new capabilities. But for now, let's just look at making more DNA. Because every time a cell completes the cell cycle and goes into mitosis, we need to have already replicated all of the DNA so that both of those daughter cells will have genomic equivalents. They will both have a complete set of the same DNA. So this process needs to happen a lot, all the time, in just about any living thing. Remember that the process is what we call semi conservative, meaning that one strand is going to be the template, and that is actually going to form the basis. It splits and that basis of each of the new strands. So we're not making two brand new copies completely separately. What we are doing instead is we are using some of the original molecule as a scaffold in order to build on. That's semi conservative. It's not conserving everything, it's building some of it new, but it is conserving some of it semi. All right, so we talked about our origins of replication, and hopefully this is pretty familiar. Remember, the replication fork is going to form, and for most organisms, oh, where did it go? Did I remove it? I guess I did. For most organisms, you're going to have lots of replication forks all over on a strand of DNA. So if we look at the DNA that makes up one of our chromosomes, we might have 10 or 20 or 30,000 different replication forks on there where um, where we can start the process of replication. Now, this is the diagram that I've included, and this is what is next in our folder. I told you that the second thing, the review, was literally a single image. This is the image. You should be able to identify each of the lettered things in here and identify not only what it is, but also what is it doing. Now, J and L tend to confuse people, but just recognize J and L are pointing to the strands. So J is saying this whole top strand is which. L is saying this whole bottom strand is which. So I'm going to kind of leave this one for the moment. You guys all have your enzymes written down and what they do. So you should be able to go back through and kind of identify everything here and what it's doing. We worked a lot on those. Remember that all of this is only ever going to work in the 3' to 5' direction on the conserved strand, the parental strand. So this is the part of the original that is unwound, opened up for replication to occur. We're looking for the 3' to the 5' of that strand in particular. You can see it labeled right down there. Now, this process is what we call anti-parallel. So the two strands are going to have to move in opposite directions because the 3 to 5 on one of them, this way, is the opposite on the other side, which you guys know influences which strand is which strand because our enzyme can only move from 3' to 5'. Now, when this process is being done, and once it is done, the polymerases are going to come back and they're going to double check just to make sure that everything's spelled right. And if there is an error, they will kind of stop, open everything up, fix and repair that error before they move on. Not always correctly, but overall, it's a heck of a mechanism. And at the end of each of our strands of DNA, we're going to have these telomeres that are produced by the enzyme telomerase. And remember, the telomeres are kind of like a timer. Basically, they're a protective cap that will stop the DNA from starting to wear away on the actual strand. It provides a section of it that can kind of be removed. A good way to think about this is if you have a beach house, your house is not literally on the ocean. You usually have 100 or 200 feet of sand. Why? If people want the ocean for their home, why isn't their house closer to the ocean? Why include that big area of sand between the two? Well, it's because that area is meant to get washed away, and yet it won't cause damage to the house yet. Your telomeres do the same thing. It's a section that we expect will get ripped, shredded, and pieces will be lost, but it should protect your house, the DNA, from getting damaged, at least for a while. Now, we also had a bunch of people who asked about viruses, and really, if we're talking about DNA replication, this might be the best time to talk about viruses as well. So remember, we're going to have things like retroviruses. And retroviruses are going to be any virus that writes its DNA into your DNA by using its RNA. So the virus is going to come in with RNA. It's going to release that RNA in, and the RNA will then be reverse transcriptioned. Basically, it will be worked into the host DNA. And then that will be replicated in red, producing offspring. Now, as we go into virus, people had a bunch of questions about them, so I wanted a chance to go over it. Now, a virus is kind of a bit of weirdness. We don't have a good grasp on what life is. I know that sounds like a dumb thing, like we should have figured that out first, but there's a lot of people that run the debate about viruses and whether or not they should be considered alive. Now, technically speaking, our best definition for life currently is that it must have all 8 characteristics of life. And the viruses fail at a couple of those. Viruses do not need to really maintain, nor do they have any mechanism to maintain homeostasis. They don't obtain or use materials or energy. But a lot of the other characteristics they have. They're basically just some form of protein coat around a core of either DNA or RNA, depending on the type of virus. And people asked about on the checklist the lytic cycle versus the lysogenic cycle. So here it is. The lytic cycle is going to be the actual production of the viruses. So the new viruses will attach to a cell, they will inject their DNA. That viral genome will then kind of take over the process. It will replicate more and more pieces of itself. The cell will then follow the viral DNA to assemble them. Eventually, the cell dies and releases new viruses, and the entire thing continues. All right, this is the lytic cycle. Now, in here, somewhere right around this area over here on the right, we have what we call the lysogenic cycle. The lysogenic cycle, as the name implies with genic, is solely concerned about the DNA. Only about the DNA and when and how are the changes being made to the DNA. Now, the lysogenic for us actually gets pretty bad because this is what allows some viruses to remain hidden, despite the fact that you have them for sometimes decades with no symptoms, no signs, you're not spreading it. They're just hiding there. They've already incorporated their DNA and now they're going to wait. Interestingly, this also will allow this lysogenic cycle will help to allow viruses that are similar to actually mix and match some of their genetic information. The best example of this that I can give you is the flu. Every year, you need a new flu shot in order to maintain your protective immunity to it. And that flu shot needs to be predicted. I think it's 2 years before it goes on sale by the CDC because that's how long it takes to make enough flu vaccine in order to sell and provide it. And when they do that, what they have to do is they have to look at what are the strains of the virus that are out right now. They have to run computer models to try and figure out what is the likelihood of them interfacing and exchanging information, and then what will the offspring most likely look at or look like. If you're not aware, believe it or not, being able to do this, they really have to predict exactly what that'll look like. And sometimes what we get just doesn't work. Sometimes what we get does not match the actual, or sorry, the expected versus the observed is different, and the shot doesn't do anything for you. All right, so that was quick, easy, dirty, but we didn't go over any of the enzymes. So, I don't think I want to do that, but I will be here tomorrow in case anybody wants to go over all of those different enzymes that are involved in replication. Because, there's a lot of them. So try to go through that diagram, label out each one, label out what its job is, what it's supposed to be doing, and then we'll check on it tomorrow. Because that's really it. There's no new other assignment today. All right, so let's see. All right, oh, yeah, let me close the recording. If you're just watching this, hopefully I'll see you tomorrow.